When we want to make predictions for the properties or functions of a protein, so far we have only looked at the protein amino acid sequences. Since we know that the protein 3D structures define the function of a protein, ideally we would like to use protein 3D structure information for this. In this video, we will briefly look at different approaches for obtaining the 3D structure for protein, and in the next video, we will look at how we can use the 3D structures to make predictions for proteins. We will start this video with a short repetition about the protein 3D structure. We know that proteins are made up of long chains of amino acids, and there are 20 different amino acids that can be present in such a chain. Each amino acid is a small molecule, and you can see an example of an amino acid on this slide, the amino acid alanine. Successive amino acids are linked together by chemical bonds, and such protein chain typically consists of a few hundred amino acids. We have already seen in an earlier video that when a protein amino acid sequence is built, the protein sequence does not remain a linear sequence, but instead it folds into a 3D structure. This folding occurs because of the attraction and repulsion between different parts of the amino acids in the chain. Additionally, some of the amino acids like to be close to water, which is typically present in the environment, and some parts try to avoid water. Because of these preferences, the protein chain bends and twists so that the water-avoiding parts are more inside, away from the water, and the water-loving parts are exposed to the water on the surface of the protein. This process is also influenced by bonds and interactions between amino acids that don't have to be close or next to each other in the linear sequence. All of these forces together cause the protein to fold into a specific 3D shape. This shape is very important because it determines the function of the protein. Here's an image that I've also shown you already before, displaying how the 3D structure of different proteins can look like. In every cell there are thousands of different proteins that all have different functions. For example, in a simple bacterium such as E. coli, which is an organism consisting of only one cell, there exist approximately 3,000 different proteins, and a total number of approximately 10 million proteins is present in each cell. All these 3,000 different proteins have different structures and functions responsible for producing all the relevant molecules for cellular growth, for taking up molecules from the environment, for producing new proteins, and for giving the cell its structure. But how can we determine the 3D structure of a protein? One option of doing this is experimentally determining it. There are different methods for doing this, which are listed on this slide. And we will shortly look at one of these, the X-ray crystallography. In the following, I will give you a very quick and broad overview over the steps involved in X-ray crystallography. First, the protein for which we want to determine its 3D structure needs to be isolated and purified. If the protein is produced by a living cell, this can be achieved by breaking open the cell and then using techniques to separate the protein from other cellular components. The second step is protein crystallization. The aim of this is to achieve that the protein molecules arrange in a regular repeating pattern in 3D dimension. Or in 3 dimensions. And once we have this, we can use X-rays to determine the protein structure. So the next step is X-ray diffraction. The protein crystals are exposed to a beam of X-rays, which is a focal stream of X-ray radiation. When the X-ray beam hits the crystals, it interacts with the electrons in the atoms of the protein. The regular repeating structure of the crystals causes the X-rays to scatter or diffract in specific patterns, and you can see an example of such a pattern in the image on the right. The next step is then to use this X-ray diffraction pattern to construct an electron density map for which complex mathematical calculations are used. An electron density map is a 3D model showing the distribution of electrons in the molecule. Rather than exact coordinates, it shows the probability of finding electrons at various positions in the 3D space, which usually correspond to atom positions. This electron density map can then be used to build an atomic model of the protein. To achieve this, a combination of the electron density information and the knowledge of protein chemistry and protein amino acid sequence is used to determine which atoms are where. And finally, there's a refinement step where the atomic positions are slightly shifted to better fit the electron density map, and the model is checked and adjusted to ensure it makes chemically sense. For example, the bond length and the angles are checked and adjusted. This whole process of experimentally determining protein 3D structures typically can take between several months and up to over a year. If experimentally determined 3D structures of similar proteins already exist, this process typically takes less time compared to determining novel protein structures. The Protein Data Bank, PDB, is the largest repository for experimentally determined 3D structures of proteins. Currently, it stores a little bit over 180,000 protein structures. And if we compare this to the number of sequenced proteins, which is over 600 million, we see that we only have experimentally determined 3D structures for a very small fraction of all sequenced proteins. We're now going to take a high-level look at how we can determine the 3D structure of proteins when we don't have their experimentally validated 3D structure. Of course, it would be highly desirable if we could simply predict the structure using sequence information. Until recently, this has not been possible. However, in 2020, Google's DeepMind presented AlphaFold 2, which is able to predict the folded protein 3D structure of many different proteins with high accuracy. This graph shows the results of the winning teams in a biannual protein structure prediction challenge called CASP. On the y-axis, you can see the GDT score, which measures the accuracy of the predicted 3D structure, with scores around 90 being considered competitive with experimentally determined structures. And you can see that in the 2020 CASP challenge, the AlphaFold 2 model that was developed by Google's DeepMind achieved prediction performance close to the accuracy of experimentally determined 3D structures. On the following slides, we will take a quick look at how deep learning models can predict the 3D structure of proteins. There have been different models published during the last few years that all achieve very good prediction results. But on the following slides, we will look at the AlphaFold 2 model, which still performs best among these models. We will not go very much into detail, but I want to give you an overview about how information is processed in the AlphaFold 2 model and how it achieves to predict all atom positions in a protein 3D structure starting from the protein sequence. First, we will look at what AlphaFold 2 receives as model input. Let's say we have a protein amino acid sequence for which we want to predict its 3D structure. First, we perform a multiple sequence alignment, or short MSA. That means we use a protein dataset to identify very similar but not identical protein amino acid sequences. This helps to define positions that are more likely to be mutated and also positions that are much more conserved and less likely to be mutated. As you can see in this example, there's positions that are more likely to be mutated than others compared to our sequence of interest in the first blue line. In addition, what an MSA also shows us is which amino acid positions co-evolve. If we have positions that co-evolve, this means that if we have an amino acid that is mutated at one position, it is also much more likely that an amino acid at another position is also mutated. This indicates close proximity in the folded 3D structure. For example, if two amino acids are close to each other and interact with each other and one is mutated, possibly maybe changing the charge of the amino acid, it may be also necessary to change the charge of the other amino acid that is close by mutating it to still get the desired interaction. Otherwise, the protein might not fold in the desired way. Finally, the MSA is embedded so that it can be processed by a transformer network. This is done in exactly the same way that we've already seen for protein transformers, the only difference being that here we have more than one sequence. But for each sequence in the MSA, we numerically encode the sequence in exactly the same way that we've seen for protein transformer network encoders, by encoding the type of the amino acid and its position in the sequence. Since we have multiple sequences, this does not result in a matrix of dimension number of residues or amino acids times hidden dimension, but it results in a 3D tensor of dimension number of sequences from the multiple sequence alignment, which is denoted by nseq, times the number of amino acids, denoted by nres, times the hidden dimension cm. In addition to the MSA, a protein structure database of known protein 3D structures is searched to find structures of related proteins. This is used to determine which amino acids in the 3D structure are likely to be close to each other and which are not. This information is then used to create a pair representation for all pairs of amino acids in the original sequence. Each pair representation has a hidden dimension CR, and we want to update this representation so it contains information about whether two amino acids are close to each other, and if so, how they interact. So this is the input for the model. On the one hand, we provide sequence information about very similar sequences from different organisms, and on the other hand, we provide structural information from similar proteins from which we already know, or for which we already know the protein structure. We're now going to look at how the MSA representation and the pair representations are updated by a newly introduced module which DeepMind called the AlphaFold. What I'm going to show is a simplification of the AlphaFold, but it should give you a good idea about how and which information flows in this network. First, the MSA representations are updated by using the attention function. Since it is computationally not feasible to apply the attention function globally to all representations in the multiple sequence alignment, the attention function is first applied row-wise. This means that each sequence in the multiple sequence alignment is updated separately in the way that we know it from the protein transformer network encoder we have discussed before. So we apply the attention function separately to each row, which is why we call this the row-wise attention. What is slightly different compared to the protein transformer networks that we've seen before is that we introduce a bias for calculating the attention scores from the pair representations. If the model concludes from the pair representations that two amino acids are in close proximity, we add a bias to the attention calculation, leading to focusing more on those amino acids that are close in the 3D structure. After the row-wise attention, there is a column-wise attention, which means the model can now look and compare the amino acids between the different aligned sequences. And it is only using this information. And to update the representations, no row-wise interactions are used between the different positions at this point. This gives an updated MSA representation, which is used to update the pair representations. For example, if we want to update the pair representations at position I and J, we use all the representations of all sequences from the MSA of the amino acids at position I and position J to calculate their interaction. This results in a vector that is used to update the pair representation. Afterwards, the pair representations are updated using a so-called triangular attention. The attention representation IJ, representing a pairwise representation between amino acid I and amino acid J, is updated using all representations between I and K and between J and K, where K can represent any amino acid in the sequence. On the next slide, we are looking at the triangular attention in a little bit more detail. After the triangular attention is applied, we have an updated MSA representation and an updated pair representation. These are then used as the input for the next AlphaFold block. In the AlphaFold 2 model, there is a total of 48 such blocks. We're now going to try to get an intuition for why we do triangular attention when updating pair representations. Let's assume we want to update the node representation between some amino acid I and some amino acid J. What we really want to integrate is the information about how those two amino acids interact and how far these are from each other. So to update the representation between amino acid I and amino acid J, we use information of the representations between amino acid I and amino acid K, and between amino acid J and the same amino acid K. This makes sense because knowing the distance between I and K and between J and K puts a strong constraint on the distance between I and J. For example, if the model figured out that I and K and also J and K are close to each other, we can also conclude that I and J have to be close to each other. By doing this, DeepMind integrated Euclidean geometry into the network. And this is done for all possible amino acids K in the sequence. The repeated application of the evoformer blocks that we've seen on the previous slide lead to an updated MSA representation and updated pair representations. Now we want to use this information for predicting the 3D structure of the protein amino acid sequence. And this is done by using a so-called structure module. We are only going to discuss very broadly here how the input is processed in such a structure module, and we are not going into much detail. The structure module receives the representation of the original amino acid sequence as its input. In the MSA, we have our original amino acid sequence of interest in the first row, so we are using these representations. Additionally, the structure module uses the pair representations for the sequence as the input. Using the structure module, we want to predict the 3D structure of the amino acid sequence, and we can do this by predicting for each amino acid its position in the 3D space and its orientation or rotation. To do this, we initialize coordinates and rotations of all amino acids. Initially, the positions of all amino acids are placed at the same initial point, and they are assigned the same rotation. The aim of the structure module is now to update these representations in a meaningful way. In the structure module, we first update the amino acid representations displayed in red by using the so-called invariant point attention, or short IPA block. This block receives the amino acid representations, the pair representations, and the rotations and translations of the amino acids as its input. As the output, we receive updated amino acid representations. These updated amino acid representations are then used to update the rotations and translations of each amino acid. Afterwards, we use the updated amino acid representations in red, the updated rotations and translations of the amino acids displayed in orange, and the pair representations that have not been updated displayed in blue as the input for the next structure module. After applying 8 blocks of structure modules, we can use the predictions for the amino acid positions and their orientations to construct a 3D model of the proteins with positions for all atoms of the protein. This slide will show you an overview over the forward process of the AlphaFold 2 model. We've seen that we first generate initial multiple sequence alignment representations where we have our sequence of interest in the first row and very similar sequences are aligned to the sequence in the rows below. We also have initial pair representations for all pairs of amino acids in the protein sequence of interest displayed here in blue. These representations are used as the input to the evoformer block that has crisscross connections between the MSA representations and the pair representations, and it updates both representations. After iteratively applying 48 blocks of evoformers, we use the updated representations for the next part of the network. Now we additionally initialize representations encoding the rotation and translation for all amino acids in the input sequence. and we use these translation and rotation representations together with the pair representations displayed in blue and the representations of the original amino acid sequence, which is the first row of the red block, as the input of this structure module. The structure module updates the amino acid representations and the translations and rotation representations of each amino acid. There's a total of 8 such structure modules, where the following module uses the updated representations from the previous module. Finally, the translation and rotation representations are used to generate a protein 3D structure with positions for all atoms in the protein. At this slide, we're looking at the final important step in the forward process. After the whole AlphaFold 2 model is applied and we have updated rotation and translation representations displayed in orange on the right side, and updated multiple sequence alignment displayed in red, and updated pair representations displayed in blue, a process is done that is called recycling. The initial MSA embeddings are updated using the updated MSA embeddings. And further, the initial pair representations are updated using the translational and rotational embeddings, as well as the updated pair representations. This makes sense because we will then be able to better integrate the information into the initial pair representation about which amino acids are close to each other. After the initial embeddings have been updated, the whole process of applying the entire AlphaFold 2 model is repeated. This recycling process is repeated 3 times, and it has been shown that this immensely improves model performance. This recycling process allows to make the network deeper without significantly increasing the training time or the number of parameters. During model training, many different loss functions that focus on different aspects are used. The main loss is the structural loss that was defined by DeepMind, which was named FABE. It is similar to a root mean squared deviation of the atomic positions between the predictions and the ground truth. Additionally, a few auxiliary losses are used. One of them is the histogram loss, which compares the pairwise distances between the predicted amino acid positions and the true distances. A further loss is used is the MSA masking, which is similar to the mask token prediction that we've seen before for protein transformers. Some tokens in the MSA representation are randomly masked during training, and a separate subpart of the model is trained to predict the type of the masked amino acids. And there are even more losses. For example, there is a violation loss term that ensures that the geometry of the predicted amino acid positions and rotations actually make sense, and that the peptide bonds between the different amino acids are in the correct positions. The model was originally trained on approximately 120,000 experimentally determined protein structures from the protein data bank PDB. After this first run of training, the resulting model was used to predict protein structures for approximately 350,000 proteins with yet unknown protein 3D structures. Afterwards, they trained a new model with the predicted structures and the experimentally determined structures as the training data, and they found that model performance improved by using this additional synthetically generated training data. On this final slide, we are quickly summarizing the capabilities and also the limitations of AlphaFold 2. We will first start at looking for which tasks AlphaFold 2 is good at. AlphaFold is good at predicting the structure of single protein chains and of protein multimers, which are protein complexes that consist of 2 or more proteins that are linked to each other. AlphaFold 2 can also be used to successfully predict the protein-protein interactions between 2 separate proteins. To achieve good prediction performance, it is not necessary to have experimentally determined protein structures for closely related proteins that can be used for training or for computing the initial structures or contact template. However, if this is the case, that no related protein 3D structure is known, it is necessary that many different related protein sequences exist that can be used for the multiple sequence alignment that is used as the model input. We are now coming to the limitations of AlphaFold 2. while, as I've just said, we do not necessarily need the 3D structure of a closely related protein 3D structure, AlphaFold 2 is typically not very good at making predictions if additionally not many closely related protein sequences exist that can be used for the multiple sequence alignment. Additionally, AlphaFold 2 is not sensitive to point mutations in the protein amino acid sequence. This is because of a lack of data on the effect of variation in the training data. And this also results because AlphaFold 2 focuses on larger patterns within the sequence and is not calculating physical forces between all amino acids and atoms, and hence single point mutations do not make a large difference when applying the AlphaFold 2 model. Proteins are also not static, but they are highly dynamic and undergo conformational changes. For example, when binding to a small molecule or performing their function, proteins are changing their 3D conformation. However, AlphaFold was trained to predict only a static 3D structure, and the prediction is hence only a snapshot of the dynamic structure of the protein. We've seen above that AlphaFold 2 can be used to predict protein-protein interactions, but it was not trained to predict interactions with other molecules such as small molecules.